psychyogi.org. Validity: the extent to which the findings and conclusions are accurate, well-founded and correspond to the real world. Now, there are a number of types of validity, which is what we're going to look at in this video. I'm not going to bother reading through all of this list now and all of this list because we're going to go through them more in depth. So, internal validity refers to the accuracy of the measurement of the dependent variable. For example, suppose an experimenter wants to measure the reaction times of a person, and to measure this they use a person with a stopwatch. Now, there's a problem with this measurement, and the problem is the person with the stopwatch also has to react. So we would expect that this measurement to be invalid, and it would be. So a better way to measure this would be to use a video camera. So we're measuring the dependent variable using a video camera of someone's reaction time. So we're filming it, and then we can watch it back and time exactly how long their reaction took. And that's a much more accurate way of doing it, and we can also repeat it. So we can have multiple people do it. And that would be high in internal validity because we have accurately measured the dependent variable, which in this case was reaction times. Now, external validity is split up into two types: ecological and population validity. External validity refers to the application to other situations and to other people. Ecological validity refers to the extent to which the results and conclusions are generalizable to real life. Ecological validity is mainly affected by the task which the participants complete. Many people say that it is the methodology. For example, a lab experiment. A lot of people will say that a lab experiment is low in ecological validity, which is not necessarily the case. You can have laboratory experiments which are high in ecological validity because the task which the participants have to do is high in ecological validity itself, so it's actually very easy to generalize that task to real life. Let's look at an example. Imagine that you are an experimenter and you are studying the psychology of shopping interactions with store clerks. So you choose a laboratory experiment because it gives you better control over extraneous variables, so thus increasing the internal validity of the study. So we set up a fake shop, which is exactly the same as one in real life. We just mirror a shop that we've seen in real life. We get a store clerk, and we tell the participants, okay, go into the store and buy some of the items that you'd normally buy. So they do this, and this is something that they would do in real life. The task they're doing, shopping, interacting with a store clerk, is very similar to that of real life. Therefore, we can say it's high in ecological validity because it's easier to generalize the results of our study and the conclusions to real life. Okay, population validity. This refers to the extent to which the sample can be generalized to similar and wider populations. Right, so the results and conclusions are included in that. Okay, this type of validity is important because without it, the research becomes low in usefulness, and usefulness is pretty much what we want for every single study we do. Let's look at an example. Suppose we're studying the impact of listening to Mozart on IQ, the so-called Mozart effect. So we decide that we're going to use a sample of people with already high IQs, and we use a random sample of a selection of people that already have high IQs. This sample would have high population validity for people with high IQs. We can generalize our results to other people with high IQs. So we're going over 100 because 100 is the average, so anything over that we're going to consider high. And anyone who has that, we can generalize our results to them and say, well, this is likely to happen given the same circumstances with this person. However, the reason it is high in population validity for that sample is because we use a random sample, which means it's more representative. Now, this sample would have low population validity for everyone else because the sample is limited. We can't actually generalize results to people who have average IQs, so most people, and people who have below average IQs. Temporal validity refers to the extent to which the findings and conclusions of a study are valid when we consider the differences and progressions that come with time. Studies that are temporally valid will either be recent studies or will be studies that consider something which has not changed since the study has been completed. For example, a study from 50 years ago into people's perceptions about height may be high in temporal validity because society's views about height haven't changed. However, Cope 1989 did a study into television advertising in the 70s and 80s, and it may not be valid today because in the 70s and 80s in England, which is where the study was conducted, there was only 3 channels. So it's not valid today because we have hundreds of channels, TV channels. We also have the internet. So we can't really generalize the results of that study to today very easily because of the changing in time, the changing in technology and the changing in people's use of that technology over time. Face validity. This refers to the extent to which a study appears to measure what it claims to measure. This is a type of validity that you're going to use very little. We typically would use something like content validity instead of it. So a study with high face validity may look like it is measuring what the researcher is intending to measure, but it does not necessarily mean that the study is measuring what it claims to measure. So let's look at an example. Suppose we are trying to measure the effects of watching a scary movie on participants' stress levels. So we show our participants a scary movie and measure their cortisol levels before and after. Cortisol is produced in response to stress, which on the surface means that this study appears as though it is actually measuring the effects of stress because more cortisol is produced when people are more stressed. So we could easily see if a person is stressed. However, we then realised that we didn't actually consider the impact of participants not eating when they were watching the film. The film was 2 hours long and they didn't eat for the entire time. So this has now caused their blood sugar levels, the blood glucose levels, to lower, which now we realise also affects cortisol levels. That will raise the cortisol levels. So our measurement of cortisol levels, although on the surface appeared like we were measuring stress, might have actually just been measuring blood glucose levels. So that actually has low content and low internal validity, but it has high face validity. Test validity is composed of several constituent parts, which we're going to go through. Test validity refers to the extent to which the results of a study or test can be said to have meaning. So let's look at an example. Suppose we are measuring the intelligence of people. We might use an IQ test. We could therefore argue, because we're using an IQ test that's established, that it has high test validity because it is a well-established test. Construct validity refers to the extent to which a study or test measures a concept which it claims to. It's further split up into convergent and discriminate validity. So, convergent validity refers to the extent to which the construct that are tested relate to each other. Discriminate validity refers to the extent to which the construct tested, which don't have a relationship, do indeed not have a relationship with one another. Content validity refers to the extent to which a study or test measures up against all elements of a construct. For example, suppose we are looking at the effects of stress on worker productivity. We have our participants answer questionnaires about how much they think they are affected by stress in the workplace and how much it affects their productivity. Now, we could argue that this is low in content validity because it hasn't actually tested the effects of stress on worker productivity. We've just asked them a question about it. A better way of doing this would be to measure how much they do and get ratings and measurements of their stress levels. The next type of validity we're going to look at is Criterion Validity, which is split up into predictive and concurrent validity. And this refers to the extent to which the results and conclusions are valid when compared with other measures. Predictive validity refers to the extent to which the results and conclusions can be used to predict real-life applications of the study. And this is not just real-life applications, but it can be used to predict what would happen in real life statistically. So let's look at an example. Suppose we are looking into the effects of thinking you are being watched on behaviour. The study is a replication of several or other studies that have been previously conducted, which suggests that there is a third chance that a person will reduce the obviousness of their behaviour. They will be more subdued in the things they do, the way they say things, when they believe they're being watched. So we do our study, we have participants complete a task. During the task, they run into several signs which say they are being monitored by CCTV cameras with operators behind the cameras. And we find again that there's a third increase. There's a third of people who subdue their behaviour somewhat. So we could say that this has predictive validity because we can see the past, there's a third, and likely in the future there'll also be that same third. Typically, predictive validity is established through repeated results over time, as we did there. Concurrent validity refers to the extent to which the results and conclusions concur with other studies and evidence. So let's look at an example. Milgram in 1963 studied the effects of obedience to authority. Milgram's results concurred with many replications of the study, and therefore Milgram's study was high in concurrent validity because we saw that the 26 participants out of 40, 63%, which reached 100% on the Volt machine, if you've seen the study, this was actually found again and again across cultures to be around 65% of people would continually comply with an authority's requests. Statistical conclusion validity refers to the extent to which we can say the results are statistically significant. That is, we can establish cause and effect above chance. Typically, in psychology studies, we have a 5% margin of chance. So whatever the results are, if they're not above 5%, then we say that chance has caused these results and we'll accept the null hypothesis. This percentage can change, and usually in physics it will be 1%, and in psychology we usually have 5%. So if it's over 5%, we can say, well, there's cause and effect here. It's not caused by chance. The dependent variable is definitely causing the change with a dependent variable, or there could be extraneous variables causing it, but we know it's not chance. Representational or translational validity refers to the extent to which the construct or concept being studied can be translated to real life. Diagnostic validity refers to the extent to which a diagnosis made about a condition is accurate. Instrumental validity refers to the extent to which the instruments used to measure the dependent variable are correct for that measurement. So for example, suppose we're measuring the length of a table and we get a tape measure and we measure it and we say, okay, this table is 2 meters long. But then we realise that the tape measure we used hasn't been accurately made, so our measurement is inaccurate, and therefore that would have had low instrumental validity. If you've enjoyed this Psych Yogi video, why not subscribe to keep up with all the latest videos.